Welcome to the Chapter 6 screencast. In Chapter 6, we are going to introduce metabolic pathways. Remember that energy is the capacity to do work. All living things must do work. Energy is required for organisms to move items within their bodies, for them to grow, to reproduce, to synthesize organic molecules, etc. Kinetic energy is energy of motion. A large truck speeding down the highway has a lot of kinetic energy. Potential energy is stored energy. A large rock at the top of a cliff has a lot of potential energy due to its position. Potential energy can also be stored in molecules. This chemical energy is due to the chemical bonds holding the atoms together. The first law of thermodynamics is sometimes called the law of conservation of energy. It tells us that energy cannot be created or destroyed. It can be changed from one form into another, and if we measure carefully, we can always determine where that energy is going. This energy doesn't just magically appear or disappear. The second law of thermodynamics tells us that no energy transformation is 100% efficient. In other words, as we use energy, not all that energy is going to go where we want it to go. The energy isn't destroyed, it's released in some other form. For example, when we exercise, we would like the food energy to go into muscle contraction, but some of it's converted into heat instead, and as a result our bodies get hot. The second law of thermodynamics is sometimes expressed in terms of entropy. Entropy is a measure of the randomness of a system. For the universe as a whole, entropy is increasing. But this does not mean that more complex organisms can evolve. Increasing structure, in other words a decrease in entropy, is possible with the input of energy. Let's try an example that you're more familiar with. You started out as a single cell, a fertilized egg. That single cell grew into your body, but that required the input of a lot of energy in the form of food. Remember, metabolism is the sum total of all the different chemical reactions that must occur in a cell. It involves all the ways that an organism acquires, transforms, and uses energy. Reactants are substances that enter into, in other words, are used during a chemical reaction. Products are what is produced. They exit from a chemical reaction. In our chemical equation, the arrow always points from the reactants towards the products. In this example, A and B are reactants, C is a product. Gibbs free energy, sometimes just called free energy, is the amount of energy released and therefore available to do work when a reaction takes place. It's calculated by subtracting the energy in the reactants from the energy in the products. If the amount of energy in the products is greater, additional energy must be added for the reaction to occur. If the amount of energy in the reactants is greater, energy will be released when the reaction takes place. Exergonic reactions produce a net release of energy. They have a negative delta G. Whereas endergonic reactions only proceed with the input of energy. They have a positive delta G. In exergonic reactions, the amount of energy in the reactants is greater than that in the products. Remember that first law of thermodynamics, that extra energy has to go somewhere. In the example reaction here, the reactant ATP contains more energy than the products ADP and a phosphate group. The extra energy is released. That released energy is available to do work. In biological systems, reactions are often reversible. A third phosphate group can be added to ADP, producing ATP, if extra energy is added. This reaction is endergonic. The amount of energy in the product, ATP, is greater than that in ADP and a phosphate group. The extra energy is stored in ATP. This reaction is endergonic. Catalysts are any substance which speeds up the rate of a reaction. Catalysts are not changed by, they're not consumed by the reaction that they speed up. Enzymes are usually proteins, or sometimes RNA. they function as catalysts. The reactant in an enzyme-catalyzed reaction is called a substrate. Enzymes are usually very specific. They often catalyze a reaction involving only one particular substrate. The active site is the location on an enzyme where the substrate attaches. If the molecule will not fit into the active site, it cannot be catalyzed in a reaction involving this enzyme. Enzymes do not make reactions occur. The reaction must be possible even if the enzyme is not present. Enzymes do increase the rate of the chemical reaction. Enzymes do not change the amount of energy in the reactants or the products. Enzymes do decrease the activation energy of the reaction. Notice in these two graphs, the amount of free energy in the reactant remains unchanged. Also, the amount of energy in the product does not change. Adding the proper enzyme makes it easier to get from reactant to product by reducing the activation energy. Environmental conditions such as temperature and pH can dramatically affect enzyme activity. Notice in this graph the rate of the reaction increases as the temperature increases until we reach a maximum value, in this example about 45 degrees Celsius. As the temperature continues to increase, the rate of the reaction decreases quickly. This decrease is due to the enzyme denaturing. The substrate no longer fits in the active site. Our enzyme cannot function as a catalyst. Enzymes are also sensitive to changes in pH. As we saw in lab, too acidic or too basic of a pH can cause enzymes to denature also. In one sense, enzymes are terrific in that they are not consumed by the reaction that they catalyze. Potentially, though, that creates a problem if we don't want the reaction occurring all the time. It's not the most elegant way to control enzymes, but potentially we could only make them when they're needed and then destroy the enzymes when they're not wanted. Another option is to control the supply of substrate. If no substrate is present, the enzyme cannot catalyze a reaction. When the product is needed, deliver the substrate and the enzyme begins to function. This option works well in eukaryotic cells, which have numerous membrane-surrounded organelles, which can be used to separate substrates and enzymes. Another control possibility is to make the enzyme in an inactive form and activate it only when needed. Adding a phosphate group to some enzymes activates them. A really cool way to control enzymes is with feedback inhibition. In this case, an inhibitor turns off the enzyme. Let's look at feedback inhibition more carefully. This diagram represents a metabolic pathway. The arrows, the individual steps along the way. A, B, and C represent molecules that are either used or produced during the reactions. E1, E2, etc. represent the enzymes which catalyze each step along the way. Reactant A fits into the active site of enzyme 1 and the pathway begins. As long as the product F remains in short supply, the pathway continues. As product F accumulates, F becomes more important as an inhibitor, turning off enzyme number 1 and thereby controlling all the reactions which follow. This elegant form of enzyme control is self-regulating. When F is in short supply, the pathway turns on. If too much F starts accumulating, the pathway is turned off. It's also sweet in that no special inhibitor has to be made. We use a product of the pathway. Finally, enzymes can be controlled using cofactors. Cofactors are any substance which is required to make an enzyme fully active. Coenzymes are organic molecules which function as cofactors. One of the functions of some vitamins is to act as coenzymes. Vitamin deficiency thus can have an adverse effect on metabolism. Photosynthesis and aerobic respiration are two examples of metabolic pathways. They're both composed of long series of chemical reactions. During photosynthesis, carbon dioxide is reduced. Electrons and energy are added as it's converted into glucose. Oxygen is a byproduct. Aerobic respiration oxidizes glucose, stripping away electrons and releasing energy. Some of this energy is available for the cell to do work. Remember, photosynthesis occurs in the chloroplasts. Carbon dioxide and water are used to make glucose, a carbohydrate, and oxygen gas is also produced. Aerobic respiration occurs in the mitochondria. In the presence of oxygen gas, glucose is split apart into carbon dioxide and water. Some of the energy that is released is captured in the form of ATP. Catabolism is any chemical reaction that tears apart organic molecules. These reactions tend to release energy. They're exergonic. Anabolic reactions involve the synthesis of organic molecules. They require energy. Photosynthesis requires sunlight energy to make organic molecules from carbon dioxide and water. It's an example of an anabolic reaction. This ends Chapter 6.